As Sonia said, my name is Lauren Altman. I am a Center for Soft and Living Matter postdoctoral fellow at UPenn, currently in Doug Durian's group. And I want to talk to you about holography and effective medium theory, which was the subject of my PhD thesis, and how we can utilize these tools to uncover the structure and dynamics of microscopic media. So before we actually get started talking about science, I want to, there we go. I want to just mention that there are a number of March for Science demonstrations going on all over the country today. And I unfortunately cannot be at the one in my city because I'm giving this talk right now. So I just wanted to throw up this slide. Here are 8 things you can do to promote human rights in science, engineering, and medicine. There's a QR code if you're interested in learning more, and please ask me for this slide if you want to include it in your future talks. So just to kind of get us oriented here, we are interested in the properties of microscopic media. So one example of microscopic media is in this video that I'm showing you here, which is milk fat globules, which are then being optically trapped to spell the word milk. And so what we're interested in here is extracting properties about this microscopic media. And so these could include the size of the colloids in this case, the optical properties, the concentration in the suspension, and their trajectories. And we can use these this raw data to answer interesting questions about the suspension that we're looking at. So for example, what are these particles made of? What is the medium made of? How do particles interact with the medium? And how do the particles interact with each other? And so the technique that we use for probing these systems is called holographic microscopy. So the way that it works is we take particles in suspension and put them in a lab slide, and then we shine collimated laser light down into this suspension. So any particles that are in the suspension will scatter the light, and these are particles that are about a micron in size. And then that scattered light gets fed through an objective into a video camera to produce a hologram like this one. And so for the purposes of this talk, a hologram, what I mean by hologram is that it's an image that encodes higher dimensional information about the scatterer. So we take this hologram, this light scattering image, and we fit it to a theory of light scattering to produce an artificial hologram, the best fit artificial hologram. And then we use that fit, which contains parameters, fitting parameters, to reconstruct a 3-dimensional object that produced the scattering pattern. So when we fit these holograms to a theory, we are typically talking about the Lorentz-Mees theory of light scattering for spheres. And so the way that it works is we model the intensity at each pixel as the sum of the incident plane wave as well as the scattered field. And this scattered field will encode information about the sphere that scattered the light, including its 3-dimensional position, its radius, and its refractive index. And I want to highlight here that this ES function is hiding a lot of complexity under the hood. So if I expand out what this function looks like, you can see that it's composed of an infinite sum of vector spherical harmonics, not particularly pretty. And the scattering coefficients are no walk in the park either. It can get quite complicated. And so the point of all of this that I'm showing you is not so that you can really absorb the form of this function. It's just to say that even fitting to the theory for spheres is quite a challenge. It's not a trivial task. But we've developed some very good analysis tools for doing this, and now we can get data like this. So on the left here, I'm showing a typical holographic characterization plot. So we can distinguish particles mixed together in suspensions by both their radius and their refractive index, which is a signal of their material, and distinguish their concentrations in this way. And we can also perform 3-dimensional holographic tracking, like this image, this video I'm showing on the right, where we are tracking a particle sedimenting and diffusing at the same time. And so when we do this technique and everything works out well, then we can get accuracy of about a part per thousand in each of the parameters that we're fitting for, which is really quite remarkable. It's very, very precise technique. So that's sort of the baseline of how this technique works. But you'll notice that up to this point, we've been describing everything as a sphere, right? So if we have a spherical particle, light will scatter off of it and produce a symmetric hologram. But in life, not everything is a sphere. So we could, for example, have some kind of asymmetric or anisotropic particle. And when light scatters off of an object like this, it will produce an asymmetric hologram. And so how do we handle asymmetric holograms like these? Well, there are a number of things you could do here. You could fit to a different exact theory for the geometry that you're looking at. And there are exact theories that exist, for example, cluster T matrix theory, but they only really exist for relatively simple geometries beyond a sphere. And you could also perform a numerical modeling. So this is a technique called discrete dipole approximation, where you would deconstruct your inhomogeneous object into a set of dipoles and then perform a numerical approximation of the scatterer. But both of these techniques are relatively inefficient, computationally expensive to perform. So what I would propose is an alternative technique to either of these, which is effective medium theory. And effective medium theory says that everything actually is a sphere. And so we're just going to take this asymmetric object and treat it as if it were a sphere and extract as much information as we can under that construction. So how it works is I have this asymmetric object, and it's made of something with a refractive index n naught, and it's dispersed in a medium nm with refractive index nm. And so what we're going to do is draw a sphere enclosing this object. And so the object will fill that sphere with a volume fraction phi. And then we'll model that enclosing sphere as an effective sphere with effective refractive index np. And then we can relate the real properties of the scatterer to these effective sphere properties using this function, which is called the Lorentz function. Okay? And so just to recap how this works, we have this asymmetric object, which produces an asymmetric hologram. Then we just fit it to the Lorentz theory for spheres as if it were a sphere. And we obtain these effective sphere properties, its axial position, radius, and refractive index. And then if we know something a priori about the object that scattered the light, then we can interpret these effective properties into real properties about the scatterer. So there's a caveat to all of this that I want to mention, which is that all of this works very well, the conventional wisdom says, if the irregularities are smaller than the wavelength of the imaging light. Okay? So that's the basic idea. And for the rest of this talk, I want to tell you about how we've applied this analysis technique to two different biological problems. One of these is molecular binding assays, and the second is the rotation of axisymmetric particles. And before we get into it, I just want to mention that this left-hand side project, the molecular binding assays, do satisfy the assumptions of effective medium theory that the irregularities are smaller than the wavelength of the light. Whereas on the right-hand side, the rotation of axisymmetric particles do not satisfy those assumptions. But we'll see that later on that perhaps we can relax those assumptions a little bit. So let's start with holographic binding assays. So this is a common technique that is used to detect target molecules in a suspension. So for example, if you had a sample that you wanted to tell if there were virus particles in there, then this would be a technique to do that. So how it works is we take a probe bead and we functionalize that bead on the surface with proteins that are designed to specifically bind to our target molecules. And so then you put those probe beads into a suspension, and if the target molecules are present, they'll bind to the surface. We can then, the conventional technique for doing this would then include an extra step, which involves fluorescent labeling. And so you add your fluorescent labels to the sample, and then you send that into a machine that can quantify the fluorescence. And if the bead fluoresces, then you know that the target molecules are present. However, we can actually circumvent this fluorescent labeling step by using holographic microscopy here. So instead of measuring the fluorescence to detect if the target molecules are present, we can actually just measure how the probe bead size changes over time. And this is a really phenomenal measurement because the size of the target molecule is really only a few nanometers compared with the probe bead, which is about a micron in size. So this is a very, very small change relative to the nominal size of the bead. But because holography is such a precise technique, we can actually perform this measurement. So you can see it working in an experimental example on the right here. So what I'm showing here are a population distribution of diameters for a particular probe bead, both before and after binding. So the dotted line or the dashed line here is this probe bead with the proteins on the surface, but before binding. And then the solid curve is after binding. And although these distributions do look quite similar to each other, there is a statistically significant shift in the nominal diameter between before and after binding. And it becomes especially clear if you look at the difference between these two distributions, which is plotted on the top here. So this is an example of it working. And what I did was I performed just simulations to see how this diameter increase would change when we change different properties of the bead and the coated surfaces. So what I did was I simulated what holograms of these coated spheres would look like using the discrete dipole approximation, that numerical technique that I talked about before. And then I take these artificial simulated holograms that I produced and fit them to the Lorentz-Mean theory for spheres to obtain effective sphere properties. And now we can see how the signal changes when we change various properties of these beads. So I'm just going to show you the results for singly coated spheres, but I can tell you that the results are quite the same for doubly coated spheres, which is more realistic to the binding assay situation that we have. And so this plot on the right here, what I'm showing is I'm keeping all other quantities fixed, and the only thing that I'm changing is the refractive index of the coating. And so that's what's plotted on the x-axis here. And on the y-axis is the difference in effective diameter between having the coating and not having the coating. Okay? And so what you can see is there's a very clear relationship here between the refractive index of the coating and the shift in diameter here. So what this tells us is that we get a much greater signal, a much greater detection difference in the shift if the refractive index of the coating is higher than the refractive index of the bead. And so this is quite important because it tells us how we can construct this diagnostic test to have the greatest signal, the greatest detection strength. So for example, these experiments were previously being done using polystyrene as the probe bead, which has a relatively high refractive index of about 1.6. And after we did these simulations, then we knew to switch to a material like silica, which has a much lower refractive index of 1.4 to perform these diagnostic tests. And I also want to mention that this diagnostic test was awarded a US patent last year for this work. So in the process of doing these experiments, we noticed something interesting going on in the characterization plots that we were finding. So what I'm showing you here is one particular experiment for this binding assay. And you can see diameter on the x-axis, refractive index on the y-axis. It's a very typical sort of experimental plot for us. And there's a very clear peak in, in this data, and that's the probe beads that we're looking at here. But then you can see that there's sort of a secondary feature in this plot. And what we believe was happening in this case was that the probe beads were using target molecules as a bridge so that they could bind to each other and form dimers. And so through this discovery, we decided to investigate what dimers would look like in the effective sphere approximation. So once again, we're going to perform simulations of holograms of dimers using the discrete dipole approximation. So we create these synthetic holograms of dimers, and then we fit them to the effective sphere approximation. And I'm just going to use the same properties of the monomers that were used in the experimental data, as I showed in the previous slide. And these were the results. So you can see the properties of the monomer as this gray dot here. And then the properties of the dimer are all of these colored dots. And I want to emphasize this is one particular dimer with one set of properties. And the only thing that's changing between all of these points here is its axial orientation relative to the imaging plane. So I don't know if you can see me, but the imaging plane is flat like this, and the laser light is coming down into the imaging plane. And so this angle theta, which is what's coloring the dots here, tells us how inclined our dimer is with respect to the imaging plane. So you can see that the blue dots over here are the dimer lying flat along the imaging plane, and the red dots are the dimer sticking straight up. And you can see that there's a very clear relationship between the effective sphere properties that you get and this orientational angle. And when you think about it for a minute, this makes sense, right? So when the dimer is lying flat with respect to the imaging plane, the sort of effective sphere, enclosing sphere looks relatively large, but mostly empty space. So a lower refractive index and a higher diameter. Whereas when the dimer is sticking straight up, the light has to pass through more material, so it looks like a smaller but denser object. Okay, so that's how you can interpret what's going on here. But really what this gives us is a way of recovering the orientational angle of a particle like a dimer just using effective sphere, right? So we can now relate what we've learned from the simulation onto our experimental data. So I'm showing you the simulation plot I showed previously on the left and our experimental data on the right. And the monomers in this case have the exact same nominal properties. And so now using this effective sphere fit as related to the orientational angle, we can then apply that information onto our experimental data and color each of our points associated with dimers with an orientational angle that's associated using a parametrized fit. So we performed this measurement with dimers. We found that you get a very similar effect with colloidal ellipsoids. And so here's that same measurement with ellipsoids, along with an SEM image of what those ellipsoidal particles look like. And if you combine all of these results together, you can get orientational distributions of populations of these axisymmetric particles. So I'm showing you the dimer plots, which are colored in orange here, as well as the ellipsoids, which are colored in blue. And I'm also including data from another group, Andreas Zottel's group, where they were looking at colloidal rods. And so in each of these cases, you find that there's a very consistent peak in this distribution here. And so really what we have here is a really consistent measure of the particle's out of plane orientation, despite the fact that each of these axisymmetric particles have very strong irregularities relative to the wavelength of light. So it's telling us that we can actually use effective medium theory even when the conventional assumptions don't hold. So we were really excited about this, and this was kind of where I left off with my PhD. And so now I'm at UPenn doing a postdoc, and I'm applying this same technique not only to study populations of axisymmetric particles, but also now to look at individual trajectories. So I am doing this work in collaboration with Arnold Matthijsen's group. And in that group, they are interested in how motile bacteria navigate through complex fluids. And so I can just tell you that certain bacteria, particularly ones that have flagella, will use shear flows to find and colonize surfaces. So this is related to a particular effect called the weather vane effect, where if a flagellar bacteria is subject to a shear flow, that shear flow will push it such that it's facing upstream. And then it will move towards the surface where the shear rate is the lowest and be able to swim upstream as a result of that. So they were interested in studying this effect, and they came to me and said, hey, can we use holography to look at the motions of these bacterial cells? And I said, sure, let's see what we can do. And so what I did is I took the confocal microscope that they already had and MacGyvered it up with a collimated laser. And so now we can perform holographic measurements with this device. And we can get images like these. So I'm going to play this video in a second, but what I'm showing you here is a hologram of a single E. coli bacterium, and it's moving through fluid in time. And we are going to record its hologram and reconstruct both its orientation in 3 dimensions and its trajectory in 3 dimensions using.
Look, Lauren, just to say, just about 5 minutes.
Okay, thank you so much.
Yeah, absolutely.
Right, so we can record its 3-dimensional orientation and trajectory in time using this technique. And hopefully this will play. There we go. And so, right, I'm just going to let this play for a minute. I'm not too worried about time, but as you can see, the sort of symmetries of the hologram are corresponding to the reconstructed cell's orientation in 3 dimensions. And so really what I want to impose upon you here is that this is a very computationally efficient technique to extract a lot of three-dimensional information about these biological media. And so finally, I want to talk about the experiments that we're running on bacterial cells like these. And so one experiment that they're doing right now is they are looking at how they can evolutionarily select for motility. So they take a particular strain of bacterial cells and they put them in the center of this agar plate. And the ones that swim the fastest will make it the furthest to the outside of this agar plate. And so then they take only the ones that made it the furthest and send that to the next cycle of evolutionary selection. So after several rounds of this, you end up with strongly motile bacteria as opposed to the original strain. So strain F is the strongly motile evolved bacteria, and strain A are the original. And using these holographic measurements, we can now uncover developments in a number of their properties and dynamics using the three-dimensional information that we can extract. So for example, we now find that strain F, the evolved bacteria, spend more time close to the walls using their axial position measurements, which of course makes sense because if they can move more efficiently, then they can find the wall more easily. They also move faster. And using the characterization data that we have, we can uncover changes in their morphology as well. So we're really excited about this and hoping to learn about a lot more using these holographic measurements. And just to conclude here, I want to say that I've shown how holographic microscopy gives us detailed three-dimensional information about suspensions with very high precision, and how we can use effective medium theory to extract information about anisotropic particles, even when the conventional assumptions don't hold, even when there are very strong irregularities. And these measurements can be applied to glean meaningful insights into biological systems. So with that, I will say thank you so much to my collaborators and funding sources, and I'm happy to take any questions. Thank you.